Okay, so let's actually walk through all of cellular respiration, including all the steps. So we know that oxygen gas goes into the lungs and it's going to be brought to cells throughout the body. So let's focus in on a cell just in the arm, okay? So if we zoom in, here's our cell. So our bloodstream is going to help bring that oxygen to cells throughout our body. So oxygen arrives at the cell. We also know that we eat food. Glucose. Awesome. So glucose goes into the stomach. It's going to go throughout the digestive tract. And as it's doing that, there are blood vessels that line the digestive tract. And they will take up glucose and send it throughout the body to all of the cells. So glucose will arrive. Okay, so we know that that goes in. Those are our two main reactants. So glucose, we'll pay attention to oxygen later. For now, let's just focus on the glucose. Glucose is C6H12O6. For now, let's just focus on the carbons. So it's a 6-carbon molecule. 3, 4, 5, 6. And the first step of cellular respiration is called glycolysis. Glyco for glucose and lysis for breaking. So what happens is we actually take this molecule and it gets broken in half. Glycolysis, glucose breaking. I'm cutting out a lot of important stuff, but this is the main gist of it. And if you remember from the last slide, some electron taxis are also filled up. Okay, and then this is all happening in the cytosol, or the cytoplasm of the cell, so the liquidy environment of the cell. But we know that the mitochondria is the organelle where a lot of the action happens. So I'm drawing it really big here. It would be a lot smaller in real life. Okay, it has inner folds, right? So we have an outer membrane and an inner membrane. And, okay, there is a step after glycolysis called the transition reaction. It's sometimes thought of as just part of the Krebs cycle, but I like to think of it as slightly different because it helps us understand how we go from outside of the mitochondria to inside of the mitochondria. But in this reaction, we take pyruvate and modify it slightly and bring it into the mitochondria. So how does it get modified? Well, 2 of the carbons stay and 1 of them gets taken off. The 2-carbon molecule that goes into the mitochondria gets coenzyme A. That's an enzyme helper. Coenzyme A put onto it. So this molecule is called acetyl-CoA, and it's what goes into the citric acid cycle, which is the next big step. This lone carbon, it doesn't just exist as a carbon on its own. That wouldn't really make much sense. Instead, this is CO2. So I'm paying attention to carbons, otherwise this would be really complicated, but remember that our original glucose molecule up there, it's not just 6 carbons, it's got other stuff in it, and that's why we're able to have some oxygens come out of this too. Okay, so glycolysis, break the molecule in half, extract some electrons. Transition reaction, we get some CO2 produced because we're taking 1 carbon off of this molecule and we're getting acetyl-CoA, which will go into what's called the citric acid cycle. This is sometimes also called the Krebs cycle, but we've talked about before how a lot of stuff in science is trying to move away from using people's last names and instead have more descriptive names. So we have our first generation of CO2. So I want to mention this is where CO2 comes from, right? Have you ever thought about you're breathing in oxygen, you're breathing out carbon dioxide? Where is it all coming from? It's coming from your cells. So this CO2 is going to leave the cell. So if our cell is in the arm there, it leaves the cell, it enters the bloodstream, it gets carried to the lungs, and then it is breathed out of the body. Back in our cell, another thing we want to recognize is that what I've just talked about here, going from pyruvate, actually I didn't write its name, let me write that down, pyruvate, our 3-carbon molecule, coming from this glucose. We just learned that pyruvate will get modified into acetyl-CoA and a carbon dioxide. But this is actually going to happen twice. Right? I'll get another CO2 and another acetyl-CoA. And they're both going to enter the Krebs cycle. Citric acid cycle. So during the citric acid cycle, we take acetyl-CoA and modify it through a series of chemical reactions. What happens are a couple things. A little bit of ATP is produced. And importantly, we fill up our electron taxis. So for every cycle, we get 4 electron taxis filled with electrons. So that would be, let's see, per turn we get 3 NADH. And this is going to happen twice per glucose molecule, right? Because we have 2 acetyl-CoAs. 3 NADH and 1 FADH. 2. And some ATP. About 2 of them. We also get some carbon dioxide generated. So a lot's happening here, but it's called a cycle because by the end of it, the last molecule that we end up with combines with acetyl-CoA to start the whole process over again. So this idea of taking our glucose molecule and breaking it down kind of ends here. By the time we go through the whole citric acid cycle, oops, I drew that arrow the other direction. Yeah, it should be like this. By the time we go through the whole citric acid cycle, we're done with glucose. We extracted everything we wanted, those electron taxis. So in here, we have many electron taxis. From one molecule of glucose, we have 6 NADH, 2 FADH2, and that's not to mention the electron taxis that got filled up earlier in this process. They'll make their way into the matrix of the mitochondria. Yeah, that's real. Calling it the matrix is the inside of the mitochondria. And then I know it's a lot, we're almost done with cellular respiration here. The last thing is now what do we do with all those electrons? And we do the electron transport chain. And it takes place throughout this inner mitochondrial matrix, right? It's not just in one spot, it's covering that entire membrane. So now I think it's going to be easier to look at an image of it instead of me trying to draw it, or we could try to draw it actually, but just know where it's happening. It's happening on this inner membrane. So maybe I'll try to give you a brief overview of what's going on here. So imagine these electron taxis, they're going to be letting go of their electrons, right? They have 2 electrons in here. When NADH, for an example, lets go of its first electron, we know it just picked up 1 free electron and generated hydrogen ions. But it had that second hydrogen that was an electron and a proton. Well, we just want to let go of the electron. So it splits it. It lets go of another proton and transfers both electrons to the electron transport chain. I mention this because this helps explain how we get all these hydrogen ions. Okay, so briefly speaking, what's going on in the electron transport chain is I'm taking electrons. And if it's not clear this first time I explain it, don't worry, we'll go over it again, or you go over it in your pre-class. Electrons get passed through a series of proteins that are embedded in this membrane. And what that does is it generates a power source. So moving electron releases energy. And what do we use that energy to do? We take these hydrogens, and those same proteins that are embedded in this membrane that are passing electrons, when they get that energy burst from passing along electrons, they're able to take hydrogen and transport it across the inner membrane. So they are creating this gradient. They are pumping these electrons out. Okay, why? Why would they want to do that? Well, the last step, I'll draw it over here, is that there are these proteins embedded separately in the membrane called ATP synthase. And what their job is to do is to passively allow those hydrogen ions to flow back into the matrix. So the thing is, these hydrogen ions are actually more abundant outside of that membrane because we have these pumps that are pushing them outside of the inner mitochondria. So what happens is these hydrogen ions want to flow down a gradient back into the inside of the mitochondrial matrix, inside of that inner membrane. The cool thing about ATP synthase is that it has this literally like a turbine inside of it. And as these hydrogen ions are flowing, it spins the turbine. It's harnessing kinetic energy in the same way that we do hydroelectric power. So for hydroelectric, crudely speaking, we put a turbine basically like in moving water, like a river. And as the water is moving that turbine, it's generating a kinetic energy that we can then harness and turn into electrical energy. So it's literally the same thing here. There's this river of hydrogen ions that flow through this ATP synthase protein. And we harness that movement energy and use it to take molecules of ADP, adenosine diphosphate, and add a phosphate to them to get ATP. And now we have created ATP inside of the cell. Okay, so that is cellular respiration in a nutshell, right? So we zoom out, it starts with eating and breathing, but we can get more and more focused. We enter the cell where glucose is broken down, and we break it down to harvest electrons that generate some CO2. We make our way to the citric acid cycle where we continue to harvest electrons and eventually go to the electron transport chain where we create a hydrogen ion gradient. And the hydrogen ion gradient is used to harness this flow of hydrogen ions back down the gradient to make ATP.